Hey everybody, my name is Brian Markowski, and today I'm going to be talking about the neurological link between social connections and physical health. The importance of social relationships in animals and humans have been demonstrated throughout history. Socially connected individuals have longer lifespans and exhibit certain health benefits, including increased resistance to certain diseases. These general benefits of social interaction are fairly well known. However, the purpose of this article was to dive into the specific neurocognitive causes of these benefits. This article primarily focuses on the neural alarm system, which is composed of the amygdala, dorsal anterior cingulate cortex, anterior insula, and perioccipital gray. The ventromedial prefrontal cortex is also featured throughout this article. The goal of this study was to investigate how social interactions activate these regions of the brain and the subsequent physiological actions that follow. Social disconnection seems to have negative health effects on humans. One theory about social disconnection is that it may be processed as a fundamental survival threat. This threat might activate the neural alarm system. In one study, viewing images of recently deceased loved ones activated this region of the brain. One of the central health risks due to the threat of socialization is inflammation. The brain can regulate inflammatory responses through the sympathetic nervous system and the hypothalamus-pituitary-adrenal axis. One of the studies referenced in this article found that chronic social disconnection is associated with increased levels of inflammatory gene expression in circulating immune cells. On the contrary, positive social interactions seem to be beneficial for someone's physical health. These positive social interactions are reinforced by reward systems within the brain, such as dopaminergic pathways. One main aspect of a positive social interaction is the perception of safety. The VMPFC is the main region of the brain responsible for safety-related processing. More specifically, the VMPFC tracks the safety value of a stimulus and inhibits the threat-related responses. In terms of social interactions, the VMPFC essentially evaluates how beneficial or threatening a future interaction will be. The VMPFC is more active during a safe social interaction or stimulus. For example, subjects that were shown an image of their supportive relationship partner during physical pain showed higher levels of VMPFC activity. I have several concerns about this research article. First, there are a lot of inferences and maybes throughout the article that suggest the authors are not confident in their findings. Also, the article relies solely on other people's studies rather than conducting their own study directly targeting their hypothesis. I thought that the article's supporting examples were not focused enough on the research question. For example, viewing images of recently deceased loved ones could result in the activation of neural regions that are involved in pain and sorrow that are not necessarily related to social isolation. I found the concept of social neuroscience to be interesting, but the methods the author used to convey this concept to be a little suspect. Thank you. Bye.